Hey everyone, welcome to today's Explainer, covering G9 Biology Unit 3, Lesson 1, Sexual Reproductions and Genetics, and specifically Meiosis. We're going to be diving into one of the most incredible processes in all of biology. We are looking at the cellular variation engine. It's literally the reason why you don't look exactly like your parents and why no two siblings are perfectly identical unless they're identical twins. It's this microscopic dance of chromosomes that absolutely guarantees diversity in life.
Okay, let's dive into this. Here's our quick agenda for today. First, the purpose of meiosis, then explaining Meiosis I and II, modeling human genetic variation, mitosis versus meiosis, a quick formative knowledge check, and finally a recap of required concepts.
So, let's kick things off with Section 1, the purpose of meiosis, the biological problem.
Let's start off with a little puzzle, okay? If you combine two typical human cells to make an offspring, why doesn't that offspring end up with double the DNA? I mean, if you think about it, the next generation would have 8 sets of chromosomes. If that just kept doubling, it would literally be biological chaos. Well, the solution lies in a highly specialized cell called a gamete. A gamete is a reproductive cell, like a sperm or an egg, that contains just one single set of chromosomes. It's deliberately packed with exactly half the normal genetic instructions just to prevent that whole doubling effect. You can kind of think of it as the essential cellular ingredient for sexual reproduction.
Now, to describe these chromosome counts, scientists use two really important terms, haploid and diploid. Most of your body cells, your somatic cells, are diploid. That just means they have 2 complete sets of chromosomes, one set you inherited from each parent. Gametes, on the other hand, are haploid. They contain only one single set. So the big question is, how does a diploid organism actually produce a haploid gamete? Well, the biological solution to reduce that diploid parent cell down to haploid gametes is an intricate process of nuclear division, and we call it meiosis.
Which brings us to Section 2. Explaining Meiosis I and II, the cellular mechanisms.
All right, so to achieve this remarkable reduction in chromosome number, a cell doesn't just divide once. It actually has to undergo one round of DNA replication, and then it's followed by 2 successive rounds of cell division. We call these Meiosis I and Meiosis II. Let's walk through this first crucial round right now, Meiosis I. Before this even begins, the cell has already copied its DNA. Then in prophase I, something completely unique happens. Homologous chromosomes, these are basically the matched pairs containing the same genes, one from each parent. They actually find each other and pair up really tightly. Moving into metaphase I, these paired-up homologous chromosomes arrange themselves right down the middle, or the equator of the cell. Then in anaphase I, the spindle fibers pull them apart. And pay attention here, the sister chromatids stay together. It's the homologous pairs that are being separated to opposite ends of the cell. Finally, in telophase I, the cell divides. Boom. We've now reduced our chromosome number by half.
But here's the thing. Meiosis I doesn't just divide the cell. It completely scrambles the genetic deck. Notice that during prophase I, when those homologous chromosomes are hugging tightly together, they actually trade pieces of DNA. We call this crossing over, and it creates entirely new recombinant chromosomes. Then during metaphase I, the way those pairs line up along the center, it's completely random. A maternal chromosome might face one pole and a paternal one the other, and it's totally independent for every single pair. We call that independent assortment. These 2 specific mechanisms are absolutely crucial because they ensure that no 2 gametes will ever be exactly alike. But we aren't done yet. Those 2 cells immediately enter meiosis II. And if you already know how basic cell division, mitosis works, this part's going to look very familiar. There's no DNA replication this time around. In prophase II, the spindles just form again in both of the new cells. In metaphase II, the chromosomes line up individually along the equator. Then in anaphase II, the sister chromatids, those exact duplicate halves of each chromosome, are finally pulled apart to opposite poles. By the time we reach telophase II and the cells divide, we have finalized our product. We get 4 completely distinct haploid cells.
Moving on to Section 3, Modeling Human Genetic Variation, the Numbers Game.
Now that we've seen the actual mechanics, let's try to wrap our heads around the sheer scale of what this means for genetic diversity. We're going to look at an analysis model here to really demonstrate how powerful this variation engine truly is. Let's play a little numbers game. 8 million. That is the number of possible genetically distinct gametes a single human can produce. 8 million. And how do we even get that number? Well, it comes strictly from the independent assortment we just talked about in metaphase I. Because the human has 23 pairs of chromosomes, and every single pair can line up in 2 different ways, the math is just 2 to the power of 23. That equals over 8 million different combinations. And remember, that doesn't even include all the countless variations created by crossing over. The likelihood of 2 gametes being exactly the same is incredibly, infinitesimally small. It's basically 0.
Next up is Section 4, Mitosis versus Meiosis, clarifying the misconceptions.
So it is incredibly common for students in grade 9 biology to mix up the two types of cell division we learn about, mitosis and meiosis. I mean, they sound super similar, right? And they share similar phase names, but their purposes and their outcomes are completely different. So let's tackle this misconception head-on. The absolute crucial point here is that Meiosis I reduces the ploidy level. It takes it from diploid down to haploid. That is a totally unique reductional event that never ever happens in mitosis. Mitosis involves just one division, and it produces two perfectly identical diploid daughter cells. That's basically just for growth and tissue repair. Meiosis, on the other hand, requires two divisions. It scrambles the genetics and produces 4 unique haploid cells purely for the purpose of sexual reproduction. Honestly, if you can just remember that key distinction, you have mastered a major, major concept in biology.
All right, Section 5, quick formative knowledge check.
Okay, focused learners, it's time to put your immediate understanding to the test. Let's just see how well we've absorbed the mechanics of this variation engine. Here is a question pulled straight from your class materials. It asks, what type of cell is produced by meiosis? Take a second. Think about everything we've literally just discussed regarding the final end product of those 2 rounds of division.
And the answer is 4 genetically distinct haploid cells, otherwise known as gametes. This brilliantly illustrates the entire purpose of the reduction division. We successfully halved the chromosome count so that fertilization can eventually bring it back to a normal diploid state, while simultaneously ensuring that the offspring is completely 100% unique. Finally, Section 6, recap of required concepts. We have covered some really fantastic, heavy-hitting biological concepts today. So let's transition into a rapid-fire summary just to cement this learning right into your memory. Here are your core takeaways. Meiosis requires 2 rounds of cell division, Meiosis I and Meiosis II. Its primary job is to reduce diploid cells into haploid gametes, which effectively solves the biological problem of doubling DNA during fertilization. Through the brilliant mechanisms of crossing over in prophase I and independent assortment in metaphase I, it shuffles all that genetic information. And the grand finale? It creates 4 entirely unique gametes. Before we wrap up, I want to leave you with a bit of a provocative thought. We've seen exactly how meiosis guarantees that every single offspring is genetically unique. But think about this. If an environment suddenly changes, say a new disease appears, or the climate drastically shifts, how might this massive variation engine actually be the key to the survival of an entire species over time? Just think about how diversity might just be nature's absolute ultimate insurance policy. Thank you so much for joining me on this explainer today. Keep learning, keep questioning, and I will see you next time.